So if a man goes to his doctor with, low testosterone, the most logical medical response is to just give him more testosterone.
Right. I mean, that makes intuitive sense.
Yeah, you're low on a hormone, you take a medication to replace it. But doing that, giving a man external testosterone to boost his levels, will completely and aggressively shut down his body's ability to produce sperm.
Yeah.
It'll make him infertile.
It is honestly one of the most fascinating paradoxes in all of human biology, and it completely breaks our standard assumption that biology operates like, well, basic plumbing, where you just pour more water into a low tank to fix the problem.
Right. Well, welcome to another deep dive. Today, we are wading right into the surprisingly murky, highly paradoxical waters of reproductive endocrinology.
Yes, we are.
We're exploring the intricate endocrine regulation of male reproduction, and specifically, we're focusing on what is happening at the microscopic level inside the testes. So I'm playing the eager specialist in training today, and you are our senior embryologist guiding us through the lab.
I am ready.
Awesome. Okay, let's unpack this. Because to understand why giving a man testosterone makes him infertile, we kind of have to figure out how this whole system is governed in the first place.
Exactly. You really can't understand what is happening on the local factory floor without first understanding the directives coming from the top down.
Right.
The entire process of spermatogenesis is governed by what we call the hypothalamic pituitary testicular axis.
Okay, so let's visualize this for everyone listening. The brain, specifically the hypothalamus and the pituitary gland, is essentially the command center. But instead of, a generic corporate headquarters, let's think of the brain as the main water reservoir and pump station for a massive, highly specialized agricultural farm.
I like that.
And the testis is the farm. The brain manages this farm by sending down two very distinct streams of water, or in this case, two specific hormonal signals, LH and FSH.
That is a perfect way to look at it. And what's crucial for you to understand is that those two streams are directed to entirely different fields on that farm. They serve completely different functions. The testis actually has a dual role, and it relies on two distinct cellular managers. So let's trace the first stream, LH, or luteinizing hormone.
Right, so LH gets pumped out by the pituitary gland in the brain. It travels down to the testis. Where exactly does it go?
So LH targets the Leydig cells, and anatomical location is everything here. Leydig cells are located in the interstitial tissue.
Wait, but before we move on interstitial tissue, we're talking about microscopic anatomy here. What exactly is that?
Good question. So think of the testes as being packed tight with hundreds of tiny coiled tubes, the seminiferous tubules.
Okay.
That is where the sperm are actually manufactured. The interstitial tissue is simply the space between those tubes.
Ooh, I see.
It's the packing material on the outside. And the Leydig cells sit in that outside space. When the LH signal binds to the receptors on these Leydig cells, it acts as the ignition switch for them to manufacture and secrete testosterone.
Got it. So the Leydig cells are sitting on the outside of the tubes, just churning out testosterone. But if they're already doing that, why does the brain need to send that second stream, the FSH? Isn't the testosterone enough?
Well, no, it's not, because testosterone alone doesn't build the physical structure needed for the sperm to grow. That is where FSH, or follicle-stimulating hormone, comes in. This signal goes to a completely different manager, the Sertoli cells. Unlike the Leydig cells on the outside, the Sertoli cells sit directly inside the seminiferous tubules.
Wait, so they are on the inside of the tubes, actually touching the developing sperm?
Exactly. They are the structural foundation. They're often called nurse cells because they physically cradle and support the developing germ cells.
Wow.
Yeah, I mean, a developing sperm cell can't even process standard glucose from energy, so the Sertoli cell physically metabolizes it and feeds the developing cell exactly what it needs, like lactate.
They are literally spoon-feeding the sperm.
Pretty much.
And they also build a physical wall, right? The blood-testis barrier.
Yes, and this is just a masterclass in biological engineering. What's fascinating here is how these two compartments work together. The Sertoli cells link together using tight junctions. It's a highly secure barricade that divides the inside of the tube into two distinct areas.
Okay.
A basal compartment and an luminal compartment.
Let me make sure I'm visualizing this right for you guys listening. Basal meaning the outer rim or base of the tube, and luminal meaning the inner hollow center, the lumen.
Precisely. So the stem cells sit on the outer rim, the basal side.
Yeah.
But as they begin to divide and mature into sperm, they have to physically pass through this barrier into the inner luminal side.
But why do they need to be locked behind a barrier in the center of the tube? I mean, what are they hiding from?
They are hiding from the man's own immune system.
Wait, really?
Yeah, you have to remember, as a sperm cell matures, it halves its genetic material. It goes from a standard cell with 46 chromosomes to a cell with 23 chromosomes,
Right.
to a white blood cell patrolling the bloodstream. A cell with only 23 chromosomes looks like a foreign invader. It looks like an infection. If that blood testis barrier wasn't there, the immune system would violently attack and destroy every single developing sperm cell.
That is wild. The body has to actively hide its own reproductive cells from itself.
Exactly.
Okay, so we have the LH signaling the Leydig cells on the outside of the tubes to make testosterone, and we have the FSH signaling the Sertoli cells on the inside to nurse the sperm and maintain this secure, hidden barrier. They are two totally separate compartments.
And they must work in absolute perfect synergy. Which, brings us directly back to the core paradox you brought up at the very beginning.
The clinical paradox of testosterone replacement therapy, or TRT.
Right.
Let's think about this TRT paradox using a thermostat analogy.
Okay.
If your house is a little chilly and you buy a small space heater and plug it in right next to the thermostat in the hallway, the thermostat senses the intense heat. It thinks, oh great, the whole house is 85 degrees, so it completely shuts off the main furnace in the basement.
Yep.
Meanwhile, the bedrooms, the kitchen, the rest of the house is freezing cold. How does giving a man exogenous testosterone, like from a gel or an injection, do the exact same thing to his reproductive axis?
Well, to understand that, you have to realize a pretty staggering fact about the local biology. The concentration of testosterone inside the what we call intratesticular testosterone, is vastly different from what is in the bloodstream.
How different?
It is up to 100-fold higher.
Wait, 100 times higher?
Yes.
How is that even physically possible? Does it just pool up like a reservoir?
Exactly. The Leydig cells are pumping it out locally, and the Sertoli cells actually secrete a specialized protein called androgen-binding protein.
Oh, so it binds it.
Right. It acts like a sponge, trapping massive amounts of testosterone right there inside the tubules. And that astronomical 100-fold local concentration is an absolute non-negotiable requirement. Without that massive local flood of testosterone, the cells simply will not undergo meiosis. They won't divide.
So here's where it gets really interesting for you listening. Because the blood tests that doctors normally run, they only show the systemic levels. They only measure the ambient temperature of the hallway, to use the analogy.
Yes. So imagine a man takes exogenous testosterone. It enters his bloodstream. The command center is constantly monitoring the blood.
Right?
It senses this huge influx of testosterone. So the brain assumes the factory down in the testes is severely overproducing, and it triggers a negative feedback loop. It immediately stops sending the LH and FSH signals.
It completely turns off the main furnace.
Precisely. Without the LH signal, the Leydig cells in the testes completely shut down their local production. So while the man's blood test might show perfectly normal, healthy testosterone levels because of the medication, his tests are suddenly starved.
Wow.
They lose that 100-fold local concentration they desperately need.
The space heater is blasting in the hallway, but the basement factory is freezing to death.
And because that massive local fuel source is gone, the entire assembly line halts. Sperm production completely stops. The man becomes infertile.
That's incredible.
It highlights exactly why looking at a standard blood test can be dangerously misleading if you don't understand the physical mechanics of the local testicular microenvironment.
That just blows my mind. The level of the hormone matters, sure, but the physical location of the hormone matters 100 times more.
Exactly.
Okay, so if this massive localized pool of testosterone and FSH is the fuel, let's look at what that fuel is actually building. If this system is perfectly calibrated, how does a cell go from a basic stem cell to a fully functional swimming sperm?
Well, it is an incredibly rigorous continuous assembly line. In humans, this entire cellular progression takes about 70 days.
70 days, okay.
And it's strictly divided into 3 distinct phases. We have the mitotic phase, the meiotic phase, and finally, spermatogenesis.
All right, let's map this out. We are going to use the strict shorthand from embryology C1, M1, C2, M2, because, tracking the exact stages is critical here so we don't get lost in the jargon. Walk us through the very beginning of the 70-day clock.
So it starts at the outer edge of the tubule, on the basal side of that barrier we talked about, with the stem cells. These are called spermatogonia. And in humans, they're classified into 2 main types, AD and AP.
AD and AP, what do those stand for?
So AD stands for A dark, because of how their nucleus looks under a microscope. Think of the AD cells as the deep resting reserve. They are the backup hard drives.
Okay.
They divide very slowly just to ensure the stem cell pool never runs out. Now, the AP cells A pale are the active progenitor cells. They are rapidly dividing, multiplying in number through basic mitosis, and eventually they differentiate into type B spermatogonia.
So during this first phase, they aren't changing their DNA, they are just aggressively building up the sheer numbers of the workforce.
Yeah, correct.
Mm-hmm.
But once they become type B spermatogonia, they do something monumental. They physically cross through that tight blood-testis barrier into the protected inner luminal compartment.
Okay.
And once they cross, they mature into a cell termed the C1.
C1 being the primary spermatocyte.
Yes, the C1, the primary spermatocyte. This is arguably the most critical cell in the entire 70-day process because the C1 cell is the one that actually initiates the meiotic phase.
And just to clarify for everyone, meiosis is the dangerous part. This is where the cell has to actively slice its own DNA in half, going from a standard diploid cell with 46 chromosomes down to a haploid cell with 23, so it can eventually fuse with an egg.
Right.
If the testosterone drops here, the whole thing falls apart.
It is an incredibly vulnerable transition. The C1 cell undergoes its first meiotic division, and we strictly refer to this division as M1.
So C1 undergoes M1.
And the physical result of that M1 division is the creation of two new cells, which are termed the C2 cells.
C2 being the secondary spermatocytes.
Exactly. Now, these C2 cells have a haploid chromosome number. They have their genetic material, but they aren't finished dividing yet.
Okay.
These C2 cells almost immediately enter a second meiotic division, which is termed M2. The M2 division separates the sister chromatids, finalizing the genetic packaging.
Okay, let me make sure I'm tracking this assembly line. We start with the primary spermatocyte, the C1. It goes through the first division, M1, to become the secondary spermatocyte, C2. Then C2 goes through the second division, M2. What actually comes up at the other side of M2?
Round spermatids.
Yeah.
Genetically, they are ready. They have the correct single set of 23 chromosomes, but physically, they just look like standard generic round cells.
Oh, those are no tail yet.
Right, no tail. They can't swim. They have no mechanism to penetrate an egg. They're functionally immature.
Which brings us to the final phase of the 70 days spermiogenesis. Not to be confused with spermatogenesis, which is the whole shebang. Spermiogenesis is just this final extreme physical makeover of the round spermatid.
It is a radical biological metamorphosis. During spermiogenesis, there is no more cell division. The cell is just being completely rebuilt. The nucleus condenses extremely tightly, packing the fragile DNA into a highly compact, crystalline payload.
Oh, so it can survive the journey outside the body?
Exactly.
It's basically wrapping the fragile cargo in armor?
Precisely. It also develops an acrosome over its head. This is essentially a cap filled with highly corrosive enzymes that it will eventually need to eat through the outer shell of an egg.
Wow.
It grows a long, functional flagellum, or tail, packed with mitochondria to act as a motor for motility. And finally, it sheds almost all of its excess cytoplasm, all the bulky cellular fluid and organelles it no longer needs.
It basically strips down all the excess weight to become a perfectly aerodynamic delivery vehicle.
Yes.
And those Sertoli cells on the inside of the tube just eat up all that discarded cytoplasm, recycling the parts.
They do.
Now, there is a really quirky, almost unbelievable detail in how these cells develop that I want to bring up. While these cells are going through C1, M1, C2, M2, they don't actually fully separate from each other.
Right.
They stay physically connected by these tiny cytoplasmic bridges. Why? I mean, why tether them all together?
It is a brilliant evolutionary failsafe. It creates what we call functional diploidy.
Okay.
Remember, after that first M1 division, the cells become haploid. They only have the genetic manual. What if one cell happens to get the half of the DNA that is missing a critical gene needed to build a tail?
You would just die.
Exactly. But because they remain connected by these bridges, the entire clone of hundreds of developing cells shares their cytoplasmic contents. They freely pass proteins, enzymes, and gene products back and forth across these bridges.
So they're literally tethered together on the assembly line, sharing tools and parts, making sure no single cell gets left behind.
Yes.
It ensures the entire cohort develops at the exact same pace.
They share the burden until the very last moment of spermiogenesis when the bridges break, and they are finally released as individual sperm into the center of the tubule.
It is a beautifully, flawlessly calibrated system. But as we know all too well in medicine, perfectly calibrated systems can and do fail.
They certainly do. And when they fail, understanding exactly where and how the failure occurs is what allows us to intervene.
Right. So let's talk about system failures. Let's say a patient has a condition called hypogonadotropic hypogonadism. Basically, the command center in the brain, the hypothalamus or pituitary, fails completely. No LH stream, no FSH stream, the farm gets no water. Is the factory permanently broken? Can it ever produce sperm again?
In that specific scenario, usually no, it is not permanently broken.
Yeah.
In hypogonadotropic hypogonadism, the physical architecture of the testes is totally fine.
Okay.
The Sertoli cells are healthy, the Leydig cells are waiting, the ad stem cells are sitting in reserve, the factory is intact, the power has just been cut. So if we act as the command center and provide those missing hormonal signals exogenously, we can turn the lights back on.
Oh, wow.
Yeah, you can often restart the entire 70-day process and completely restore fertility.
Okay, but what if the opposite is true? What if the brain is sending the signals, the factory is working perfectly, the 70-day assembly line builds healthy sperm, but there's a problem once the sperm leave the building? How does a man end up with severe male factor infertility if the testis is working?
This is where we step into the realm of ART-assisted reproductive technology.
Right.
Historically, if the sperm were being made but couldn't safely exit the male reproductive tract due to a blockage or severe damage during transit, there was nothing we could do. But today, ART allows us to physically bypass the entire excurrent ductal system.
So we just go in and get them. And the procedure mentioned here is TESE, testicular sperm extraction. How exactly does TEC bypass these roadblocks?
So TEC involves a minor surgical procedure where we retrieve microscopic amounts of tissue directly from the testes itself. We're plucking the sperm right out of the seminiferous tubules rather than relying on an ejaculated sample that has traveled through the body.
But why go to the trouble of surgical extraction if the man is producing sperm that technically make it out, but are just, poor quality?
It fundamentally comes down to the quality of the genetic payload, specifically a metric called SDF, or sperm DNA fragmentation.
Okay.
You see, the sperm are manufactured perfectly well inside that highly protected, immune-privileged environment of the Sertoli cells. But once they are released, they have to travel through the epididymis and the entire male excurrent ductal system.
Which is a long way.
It is a long, winding journey. And during that transit, they are highly vulnerable to oxidative stress.
What is oxidative stress actually doing to the sperm? Is it just like tiring them out?
It's far more destructive than that. If a man has minor inflammation, perhaps from an enlarged vein like a varicocele or an old infection, white blood cells in those ducts release reactive oxygen species free radicals.
Oh, I see.
Because the sperm shed all their protective cytoplasm during spermatogenesis to become aerodynamic, they have virtually no cellular defenses left. These free radicals physically attack and shatter the condensed DNA strands inside the sperm head.
Wow.
It is essentially rusting the genetic cargo from the outside in.
So the transport journey itself is a gauntlet that destroys the cargo.
Exactly. By utilizing TESE, we capture the sperm directly at the source right off the assembly line before they ever embark on that damaging journey. We retrieve a sperm with completely intact, unfragmented DNA.
But there is a catch, right? Because sperm pulled directly from the testes haven't gone through the epididymis, they aren't fully mature swimmers yet, they can't move well. You can't just put a testicular sperm in a dish with an egg and hope they figure it out. You have to pair TEC with ICSI, intracytoplasmic sperm injection.
Yes, the two procedures are inextricably linked in this scenario. We take that single, genetically pristine testicular sperm retrieved via TESE. Then, under a high-powered microscope using a microscopic glass needle, an embryologist physically injects that single sperm directly into the cytoplasm of a mature oocyte.
You inject it right into the oocyte.
Yes. By combining TEC and ICSI, we completely bypass two massive biological hurdles: the oxidative damage of the male tract and the rigorous natural selection barriers required to physically penetrate the oocyte.
That's amazing.
For men with highly fragmented DNA in their ejaculate, it can be an absolute game changer.
It's incredible. Modern medicine is literally stepping in to act as the transport mechanism and the fertilization trigger. Well, okay, we have covered a massive amount of ground today. Let's pull all of this together for you.
We started at the hormonal command center in the brain and ended at the microscopic injection of a single cell into an oocyte. It is a long journey.
To distill this down, spermatogenesis is a profoundly delicate, highly coordinated dance. It relies on a critical balance of two streams from the brain. FSH acts on the Sertoli cells inside the tubes to nurse the sperm and maintain the blood-testis barrier.
Right.
And LH acts on the Leydig cells outside the tubes to manufacture testosterone. But the crucial defining takeaway is that the local concentration of testosterone inside the testis has to be up to 100 times higher than what is circulating in the blood.
Exactly.
Standard blood tests simply do not tell you what is happening on the factory floor, which is why taking external testosterone tricks the brain and shuts the whole local system down.
And if that that system is running perfectly, it is a rigorous 70-day cellular progression. It moves from C1 to C2 via the M1 meiotic division, and then to round spermatids via M2. All of this culminates in the drastic physical remodeling of spermatogenesis, where the cell strips away its excess baggage.
And when that system hits a roadblock, whether from hormonal failure at the command center or oxidative damage during transit, modern ART gives us tools. Using TSC to extract the sperm at the source, combined with injecting it directly into an oocyte, allows us to completely bypass the damage.
It truly highlights the resilience of human biology and how elegantly we can intervene when we actually understand the underlying cellular mechanisms.
Which brings us to our final thought. Throughout our entire discussion today, we have focused exclusively on the male side of the equation. But if you step back and look at the broader picture of human reproduction, there is a stark, fundamental difference between how male and female gametes are built. And it's something I want you to seriously think about.
Yes, it is a biological reality that forces us to reframe how we view fertility. We established today that male meiosis and sperm production is an ongoing, continuous 70-day cycle. It is a factory that never closes. It restarts constantly throughout a man's adult life. Millions of brand new cells are built from scratch, undergoing complex genetic halving every single day.
But female meiosis is an entirely different biological strategy. A female's oocytes actually begin their meiotic division while she is still a fetus in utero, and then they arrest.
The hip pause.
Right, they sit in that suspended state of animation for decades, only resuming the process one by one, month by month, after puberty.
So I want you, the listener, to consider what this fundamental difference means for environmental vulnerabilities.
Oh, wow.
If male gametes are constantly being constructed, constantly dividing their DNA, and differentiating in a highly sensitive local microenvironment every 70 days, they are essentially a real-time sponge for whatever environment the man is currently in.
Right.
How might modern lifestyle factors, daily chemical exposures, microplastics, or even minor temperature changes uniquely shape the fragile genetic payload of a spermatozoon being built on any given Tuesday?
Compare that rapid, real-time construction to an oocyte that has been resting largely shielded and locked away in a protective vault in the ovary for 20, 30, or 40 years. It completely flips the script on how you might view a simple daily habit. Is the continuous, hyperactive factory actually vastly more vulnerable to today's modern industrialized toxins than the decades-old vault?
It is one of the most pressing questions that reproductive endocrinologists and embryologists are actively wrestling with right now.
And just like we discussed when we first looked at the paradox of testosterone therapy, the deeper you look into the muddy, complex waters of biology, the more you realize that a simple snapshot never tells the whole story. You always have to look at the local environment. Thank you so much for joining us on this deep dive. Keep asking questions, keep looking beyond the surface, and we will catch you next time.